if we cease adding antibiotics to the food of animals, we are going to experience very significant loss of profit, but also some people are claiming that there will be lack of protein, which I believe is extreme, but there is a lot of controversy about why and how we have to use antibiotics. We understand why we don't have to use antibiotics so widely because of antibiotic resistance. This is a chart that is giving you an idea about what kind of antibiotics are used in food production. This is from Holland. In Netherlands, they are using a lot of tetracycline is the green bar here, the blue bar underneath is penicillin. These are other microbials. You can see that it was steadily growing until 2007, and then there is a sharp decline. The sharp decline is dictated because of these fears of antibiotic resistance. And because there are legislations that are actually going to enforce reduction and phase out of antibiotic use in livestock production. In USA, I know that there is a law that passed about a year ago, something like that. European Union is following suit. Canada will as well. This trend will continue. We know why. Antibiotic resistance, a few facts for those of you who like facts, projects. This is very important review. That's a review commissioned by the government of United Kingdom. And people figured out that by 2050, if we don't have a grip on antibiotic resistance, we can have 10 million deaths per year because of antibiotic-resistant bacteria, which is quite scary. Quite scary. It makes good politics, by the way. By any measure, there will be reduction of use of antibiotics, meaning that there will be a reduction in productivity of livestock. So we have to find something to substitute for all this stuff. And the substitution that we came up with, and other people have come up with, are alternative antimicrobial agents. And one of the most powerful and actually established by nature defensive mechanisms are these molecules which are called defensins. Defensins are widespread. They are in our cells. They are human defensins, mammalian defensins, plant defensins, and fungal defensins. Every species has a defensin of one kind or another. The very interesting feature is that all these defensins share the ability to very quickly and efficiently kill bacteria. Very quickly and efficiently, more efficiently than antibiotics. They are expensive, though. They are expensive to produce. I told you that they are widespread, that many of them disrupt membranes. We know this. But many of them also have an additional function. And this plurality of mechanisms, antimicrobial mechanisms, confer these little anti-defensin defenses of the microbes. Microbes do not have readily available genetic system to combat defenses the way they have for antibiotics. So we know if you have an antibiotic, antibiotic resistance would be conferred by an enzyme that is passed horizontally through different species. There is nothing like that against defensins, which makes them very attractive candidates for substitutions of antibiotics. So defensins, let's decipher what a defensin is. Not usually, these are small molecules, positively charged peptides, less than 100 amino acids. The ones that we know best are about 50 amino acids, and they are characterized by structure. Their signature is a structure. The structure is conferred by 3 to 4 disulfide bonds, which are depicted here in yellow. And we have a structure that I will not describe to you because I'm not a structural biologist, but we can picture how this thing folds and confers a cluster of positively charged amino acids that will touch the negatively charged membrane. And then there are other activities that we don't quite understand. So, defenses. They are everywhere. Why fungal? Why fungal defenses and not, for example, human defenses? Well, human defenses also communicate with the immune system. So their continuous use and overuse may actually cause deleterious effects, and actually they do. There is documented immunosuppression, immunoactivation by different defenses. So we have to be careful and watchful when we propose defensins as for use in our environment. So we don't want to eat defensins, human defensins. We want to do something else. Fungal defensins are known, well, are not known is the right word, are not known to cause any side effects in mammals. So that's good news. We treat them as food, as something that's out there. We don't have physiological response to these defensins. There are only 14 of them identified so far from fungal species. And this is where my hope is. If they are so good, if they are so inert in terms of human physiology, can we just develop a system to defend against microbes by producing some of these proteins? And some of them have been actually produced, patented. Some of these are patented and are poised to be used in production. What is our idea? Our idea is to make them affordable. Everything that I described so far about defensins is true, except that we don't know how to produce them in large quantities. We also don't know how to isolate them. I still have a little bit of time. Of these 14, only 3 have been identified by routine methods. That means grow fungal culture, harvest this culture, purify the proteins, isolate the activity. The other ones were identified by mining genome databases, like, you know, we sequence everything these days, so whatever is sequenced out there, people have looked for these cysteine bonds, and 14 of these have popped up, and that's how we look for them. There must be many more defensins that we do not know, we do not identify, and the process of finding defensins is actually quite expensive, and their production is also expensive. So right now, I think the limiting factor is financing these projects. Antibiotics are affordable. Oh, another cool feature of defensins, these are peptides. When we ingest them in our stomach, they get degraded, they're gone. They're not going to affect the microbiome of the intestines of our gut or the gut of the animal, whereas antibiotics do, big time actually. When I was reading about preparing for this talk, I was shocked to find that 75% of the antibiotic that is given to livestock is found in feces, in the farms. The farms are actually a huge depository of any kind of antibiotics. So this is a bad feature. Again, this is not going to happen with defensins. They are going to convert into food right away. So this is the strategy that we came up with. The strategy is to generate a library, to use genetic means, to generate a library of fungal-like defenses that might or might not exist out there in nature. And we figured out that we can handle up to 50 million at a time, 10 to the 7, 10 to the 8. That's a big leap forward. If you remember during the first talk by Dr. Friese, he said that they can handle up to 5,000. We are talking about 50 million. Genetics is more powerful. Everybody knows that. Genetics has power. What are we going to do with this library? We are going to put it in budding yeast. Budding yeast, many of you know, is a fungus. It's a fungus, so a fungus expressing fungal proteins, that's quite good. And then we are going to do this tricky assay that we call confrontational screens. That means that we are going to mix our library with the target bacteria and look for activities that are expressed by the yeast and kill bacteria. After that, we isolate these genes and put them in another yeast. That's Kilberomyces lactis, K. lactis I always pronounce it. This is a tongue twister. It makes kefir. It's food. This is making food. It's not something miracle. So kefir, we are going to feed instead beer. We are going to give cows kefir, and they're going to get healthier. So that's the idea. Let's go through the strategy in a little bit more detail. Why S. cerevisiae and not K. lactis right away? Because we can handle S. cerevisiae better than K. lactis. S. cerevisiae is a model organism. It's been around for quite a few decades, genetically very, very well developed, and we have the expertise. We can do genetic screens in S. cerevisia way better than in K. lactis or any other eukaryote. That's arguably the best developed, some call it almost eukaryote, but I call it eukaryote. Okay, so it's a good organism to study. Why synthetic? Because of all the arguments, because of all the difficulties that are associated with starting with natural sources or genomes. Okay? We are going to propose that these defenses exist and produce them and then select. What kind of library? I'll tell you what kind of library. I explained why we can handle these many clones. That's simply how many colonies we can handle at a time. Basically, the limitation comes to how big is your incubator, how many plates you can put in your incubator, and we figured out that we can put 100 plates containing this many clones. We are working on a strategy to go to a higher number of clones, but that's for the future. Then express, why then express in K. lactis? I told you, we are in Germany, in many countries you can be prosecuted if you give beer to cattle, and it's actually not a good idea. Beer is for us, it's good, we're going to enjoy beer. Cattle are going to eat kefir, things like that. The real reason is that K. lactis is a way cheaper system to express proteins. Milk whey, that's a byproduct of cheese production. It is really affordable, and there would be no considerations whatsoever that this is food and nothing else. More details, we have a vector. We've made a vector. We synthesized this vector from a company called DNA2. They make it for very little money, actually. $500–600 is the total cost of this vector. The key to the expression, the key to this vector is the alpha factor peptide. This is a peptide that guarantees that your protein is going to be secreted. The trick is to get it out of the cell, not in the cell. In the cell, we don't need it. So this is where we are struggling a little bit, but I'm not going to share with you our problems. We don't get enough extracellular expression at this point. We have to improve this. We will play around with this leader peptide. But that's everything. The rest is selection markers, promoters, things like that. We have promoters. The protein will be expressed, okay? That's that's almost given. The library. The library, if we have 35 amino acid peptide, we are going to get 35 to the power of 20th clones. That's a big number. There is no way that we can handle these many colonies. So we have to do something smart to bring it down to this range, 50 million, and what we are doing here is limited degeneracy. So we know that at every position we can have one of 20 amino acids. We have to do something, and the first thing is no-brainer. We are going to conserve the cysteines. This is codon 13 will be cysteine, 17 will be cysteine. That's a real situation. That's our library, actually. It's not something for, it's not an example for you. These are the positions of cysteines. We also decided to conserve this isoleucine here, something hydrophobic, okay? It's not always isoleucine, but we decided to put isoleucine because it looks like the other ones, histidine. And then we provide limited degeneracy at different positions. At 11 and 12, we say we are going to go for a charged amino acid. Because over there we have positive, negative charges that alternate. So at these positions, we are going to go for a charge. At this position for something else. By these tricks, some people call it intelligent design. I don't think that it's intelligent in this situation. It's desperate design. We have to reduce the number of clones, and this is what we come up with. If we are successful, I'm going to show you the cost of this limitation. We'll come to money later on. Confrontational screens, I developed a cartoon for you yesterday evening while not sleeping. Jet lag, you know, things happen. First step, get all these plasmids, generate a library that would represent 10 to the 8 clones in budding yeast. Then plate the budding yeast on a plate. Start growing the budding yeast, and at the same time or right after, you're going to flood this plate with the bacterium that you want to confront. And if you are lucky, if you have a molecule that is suppressing, killing these bacteria, you're going to see something that is not very well seen here. You're going to see a clearance on the bacterial culture around the colony of yeast. It's an old trick. Okay, that's how antibiotics were discovered. Okay, you grow one culture next to another culture, and if one culture kills the other one, you're going to see a clear area in the plate. So this is what we are aiming to do, and we have achieved something like that. If you don't have, I don't know how to switch off the light. If you have yeast cells that do not express the library, you can see them as yellow remnants on the plate. This is all flooded with staphylococcus over here. On this plate, you can see a tiny clearance over here, which is enlarged over here. So it is possible to do this. That's all that we have achieved so far. This is a feasible procedure. However, this clearance is not at all the tool that we want to start screening a library with. We have to improve the whole system. We have to solve a couple of problems: better expression, better excretion, I would say, of the defensins, so we have better activity outside the cell. This is very low activity. That's minimal. We have to figure out how to co-culture bacteria and yeast. Yeast are slow growing, bacteria are fast growing. There are technical issues, but I think they are within our reach, these technical issues. Here is the cost of the project. Generate the library. Intelligent design, intelligent input, and a little bit of money would achieve that. Actually, this has been achieved. We have synthesized the library. We have made it degenerate. The vector is $500. Another company is synthesizing the library, cloning it. That's cheap. That's cheap. Expressing budding yeast, $7,000 is the stipend of a student. That's all, the stipend of a student for the summer. That's our expense. Anything else is consumables that are available in the lab. We are at this stage. We have to develop a powerful, reliable confrontation assay, and then to conduct the screen, and that would be a little bit more expensive. But when you look at it, with all pros and cons, within, say, $50,000 to $100,000, we'll be able to conduct one screen, to complete one screen, which is not horribly expensive when you think about that. I will stop here. I will stop here because we started late and we want to have lunch at the right time. Money-wise, I explained how the situation looks like. I want to acknowledge two students who are have been working on this project: James Haskins and Brandon Wise, undergraduate student and a graduate student. The two of them have been working on this project. The third one, Prithif, is coming this fall to join them. Acknowledgements for the funding agencies, and CIRC is giving money for reagents, and stipends are provided by these two guys. And at this point, I will thank for your attention and be prepared to answer your questions. Thanks for your attention.